Ladies and gentlemen, may I please have your attention as we are about to begin. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, may I please have your attention as we are about to begin. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, may I please have your attention as we are about to begin. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Oktober tahun ini dong, 2022 dong, 2003 dong. Ladies and gentlemen, may I please have your attention as we are about to begin. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Good morning and best wishes us all. Our Honorable Chairman of Research Organization for Agriculture and Food BRIN, Mrs. Puja Lestari, PhD, who is also today's speaker. Our Honorable Speaker, Dr. Mulyana Hadi Pernata, Head of Research Center for Agro-Industry BRIN. Professor Ichiro Fukusaki, Principal Investigator of COI Next Project, Osaka University, Japan, and Dr. Sastia Pramaputri, Associate Professor, Osaka University, Japan, and our moderator for today, Mr. Andi Febrisiantosa, PhD, Researcher of Research Center for Food Technology and Processing BRIN, and all participants this morning. First of all, let's be grateful to Allah Subhanahu wa Ta'ala, who has given us good health and time so we can attend and participate in the sharing session presented by Research Organization for Agriculture and Food BRIN with the topic Reduction of Food Loss on Tropical Fruit. Wow. Next, we would like to invite the Chairman of Research Organization for Agriculture and Food BRIN to give welcome speech and open the sharing session. For Mrs. Puja Lestari, PhD, time is yours.
Thank you, Ms. Warda. Good morning, everyone. Could you hear my voice?
Yes.
Yes.
Okay, thank you.
Clearly.
Yes.
Okay, thank you.
Okay.
Yeah, thank you so much.
Clear.
Yeah, it is a glorious moment to extend my warm wishes on behalf of Research Organization for Agriculture and Food, BRIN. I would like to convey my heartfelt gratitude to distinguished speakers from Osaka University, Japan, Professor Ichiro Fukusaki and Dr. Sastia Pramaputri. My gratitude is also addressed to scientists from BRIN universities and other research organizations from Indonesia, Japan, and probably from other countries who have made it to this first session with topic of reduction of food loss on tropical fruit. Ladies and gentlemen, food loss has negative environmental impact because of the water, land, and other natural resources used to produce food that no one consumes. The size of the impact increases with the level of processing and refining of the food products and the stage in the food supply chain at which the quantity and quality of food are lost. Generally, lower losses are associated with higher efficiency in the food supply and eventually more effective recycling of resources, lower storage needs, shorter transport distance, and less energy use. Post-harvest loss of horticultural crops exacerbates humankind's agricultural problems and will continue to do so in the next decade. Tropical fruits provide us with a vast spectrum of heartful nutrients and enrich our lives with a wide array of pleasant sensory experience. These commodities are, however, highly perishable. Approximately At least 30% of the harvested produce is never consumed, since these products naturally have a short shelf life, which leads to post-harvest loss. Indonesia has diverse tropical fruits across the country, and they play an important role in society's economic and social aspects. However, some fruits have a relatively short shelf life. Those post-harvest packaging and handling strategies are necessary to achieve optimal fruit maturity and maintain quality. A better understanding of the strategy of reducing the loss of fruits in the process of on-farm and off-farm could help design precision technologies. Therefore, a research strategy on tropical fruits is needed to reduce food loss along production and supply chains. Research Organization for Agriculture and Food, other National Research and Innovation Agency, Republic of Indonesia, BRIN, highly concerned about reducing food loss of fruit in the country. Therefore, collaboration research to boost the achievement of goals on this research and innovation are being initiated by BRIN and Osaka University, Japan. And today's first sharing session on reduction of food loss on tropical fruits is part of sharing session series organized by BRIN in collaboration with Osaka University. We're supposed to have 2 more webinars in this June. Hope it could be beneficial to increase our knowledge on this topic, reduction of food loss, and to discuss a brief concept note of the research project proposed by BRIN, especially Research Organization for Agriculture and Food, and to strengthen networking and initiate collaboration between BRIN and Osaka University. So today, this morning, with Bismillah, we open this sharing session, and I wish and I ensure that this sharing session will be profitable and fruitful for everyone present here. Thank you very much. Mbak Wardah, that's all for my speech. Thank you.
Next, we would like to invite all participants to join a photo session. For all participants, please activate their videos. May I ask Ms. Mala Dewi Agisti to please help with documentation. Please, Ms. Mala. Thank you. Ladies and gentlemen, without further ado, this meeting will start soon and will be guided by our moderator. For Mr. Andi Febrisiantosa, PhD, time is yours.
Yes, thank you so much, Mrs. Wardah. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. The honorable speakers and the honorable participants of today's event. I'm Andi Febrisiantosa, as already mentioned by the host, and I'll be a moderator for today's webinar. Hello. Could you please hear clearly my voice? It's okay?
It's okay.
Okay, yeah. I will be moderating of the event, and I'm very pleased to see you all here and welcome all of you to this sharing session webinars today. So today's sharing session is about reduction of food loss on tropical fruits. Ladies and gentlemen, before we get started, I would like to inform you a few items so you all will know how to participate in today's event. We will have 4 presenters, and I would now like to inform you our presenters today. So we will have presenters from both of sides, from BRIN as well from Indonesia, as well as from Japan. The first presenter will be from Professor Eiichiro Fukusaki, a principal investigator principal investigator of COI Next Project, Osaka University, Japan, with the topic title Food DX Technology to Reduce Food Loss and Food Waste. And the second presenter will be from Dr. Sastia Pramaputri, Associate Professor from Osaka University, Japan, with the topic Metabolomic Approach to Extend the Shelf Life of Tropical Fruits. And the third presenter will be Puji Lestari, PhD, the chair lady of our research organization, Research Organization for Agriculture and Food, BRIN, Indonesia, with the title Overview of Research and Reduction of Food Loss on Selected Tropical Food in Indonesia.
Ini bukan 500,
and the fourth presenter will be Dr. Mulyana Hadipermata, the head of Research Center for Agro-Industry, BRIN, Indonesia, with the title Profiling and Sensory Evaluation to Support Post-Harvest Handling and Agro-Industrial Processing of Tropical Fruits. Firstly, we will have all of presenters will be invited to deliver the presentation, and after that, we will go to the question and answer session at the end of this today event. After all the presenters deliver their material. And for the audience, we will have opportunity to ask questions directly by raising a hand or put the raise hand symbol on your panel during the session answer question session. And you will also have to the opportunity to send the question by typing your question into the question pane of the control panel. And you also may send your questions at any time during the presentation. We will collect this and address them at the end of the presentation. Okay, ladies and gentlemen, now let's please, I welcome our first presenters and say hello to Professor Eiichiro Fukusaki. Hello, Professor Eiichiro.
Hello.
Have you been here? Hello. Yeah.
Hello.
Yeah. Good morning here in
Good morning.
Indonesia as well. Maybe in Japan also still morning, right? Yeah, we have different 2 hours, but it's okay. Yeah, our pleasure having you here, Professor Fukusaka. In this session. Let me introduce first Professor Fukusaka profile. Please show the Professor Fukusaka profile. Yeah, Professor Eiichiro Fukusaki, Eiichiro Fukusaki is a principal investigator of COI Next project from Osaka University, Japan. And area of his expertise is metabolomics, metabolomic profiling, bioengineering, and metabolomic fingerprinting. He finished his PhD in Osaka University in 1993, and the last decade or the last his work and team is work related to the metabolomic approach to elucidate the importance of concise in commercial pennies shrimps, yeah, in the source shrimps and several products related to the fresh product, food product. Yeah, so there's no question of his expertise, and let's see his presentation about food DX technology to reduce food loss and food waste. What is food DX technology and how it rules to reduce food loss and food waste will be presented by Professor Eiichiro Fukusaki. So please to deliver your presentation, Professor. The floor is yours. Thank you.
Yeah. Good morning, Indonesian friends, and thank you very much for your kind introduction. So it's my great pleasure to have an opportunity to speak my research activity. So can I share my screen?
Yes, please, Professor.
Can you see the screen?
Yes, very clear, Professor.
Okay,
Yeah.
let me start my talk. I'm at Fukusaki Osaka University. By the support of the Japanese government, we have launched a research center hub to solve the problem of food loss reduction through technological innovation by a combination of the latest technology from fiscal year 2021. According to the FAO report, as you know, about 1/3 of the world's food production or enough food for 2 billion people is wasted, resulting in annual economic loss of 80 trillion Japanese yen. On the other hand, the annual carbon dioxide emission from food loss is 3.6 billion tons, which is 8% of the world's total. Considering that China, that world's largest emitter of carbon dioxide, emits 9.6 billion tons per year. The United States, second, India, the third, emits 2.3 billion tons. We can see how important an environment problem food loss is. Among the food loss, production loss, storage loss, processing loss, and distribution loss can be reduced by advanced technology. Furthermore, in order to reduce consumption loss, which are common in developed countries, it is necessary to change the mindset of consumers. With a vision of realization of low food loss, is a consumer society opened up by Food DX.
Sorry.
We would like to tackle the extremely serious and global social issue of food loss. Our center will be located in Japan, but we are considering Indonesia as a most important test field to prove the concept. First, let me explain why Indonesia. Indonesia is the second largest food loss country in the world. At the same time, Indonesia is the largest fruit producer in Southeast Asia, with an annual production of around 20 million tons. The figure in the middle shows the breakdown of food loss in Southeast Asia and in the industrialized region of Asia. In South Asia, storage, processing, and distribution loss are very high, and 44% of the food is wasted. On the other hand, in developed countries such as Japan, storage, processing, and distribution loss are limited to 80%. If the storage, processing, and distribution loss in Indonesia could be improved to the level of Japan through technological innovation, it would lead to a 26% reduction in food loss, theoretically. And the annual reduction in food loss from fruits alone would be about 5 million tons. This amount is equivalent to 0.3% of the world's food loss, and the economic effect would be about 240 billion Japanese yen per year. Okay, next we backcast the solution strategy based on the 5 million ton of food loss reduction as the future. This project consists of 5 research and development projects, including a social science project, building a sustainable socio-economic system through ethical consumption, and 3 emerging technological project development of rapid monitoring technology for food function, development of smart data logger system, and development and utilization of zero waste recycling food material, and social science and humanity project sustaining human resource for food solution and creating opportunity through interactive international education program. In this way, 5 research and development projects will be connected to form a functional cycle of social implementation of the result of further challenge to new target. In the effort to reduce food loss by 2031, 10 years from now, we hope to establish a resilient food supply chain and reduce food loss from current 33% to 18%. As a result of emphasizing diversity, more than 1/3 of the members are humanity researchers and more than 1/3 are female researchers. In addition, since this is a long-term project spanning 10 years, the majority of the members are young researchers in their 30s and 40s. I would now like to explain about each R&D task. Research and development task 1 is to develop a rapid monitoring technology for food with the concept of cattle food. In this project, I, a leading expert in food metabolism analysis, has already succeeded in rapidly and quantitatively measuring the value of food function and freshness. By further developing this, we will develop food gas metabolism technology and build a model for predicting food function deterioration. Furthermore, we will develop real-time monitoring technology through collaboration research. This research project is connected to the next R&D task 2. In R&D task 2, we will develop a smart data logger system based on the concept of lab food. Under the leadership of Professor Hirose, who is a leading expert in integrated electronics technology, this project will integrate the result R&D task 1 into a chip using food DX technology, and will integrate automatic food data collection system technology and food gas sensoring technology. In addition, in order to prevent food fake, we will attach an ultra small tag, the food, which records all the data of the food in the distribution process in a form that cannot be tampered with to ensure its authority. This tiny piece of equipment is the world's smallest computer. By attaching this computer to individual foods, we are developing food security and safety technology that makes the history of food completely visible. Task 3 deals with the development and utilization of zero waste recyclable food material based on the concept bring up food. In this project, we will develop reversible freezing drying technology that preserved and restore the original characteristics of food. Led by Professor Uchiyama, a leading expert in stabilization technology for biologic pharmaceutical protein such as antibody and vaccine. This technology minimizes the distribution of food at low temperature and realize a decold chain. In addition, Professor Muranaka and his colleague, who are leading experts in genome editing technology for practical plants, will play a central role in this project to make effective use of plant and microbial resources that have not been used so far. In addition to the leader, Professor Muranaka and Professor Uchiyama, Professor Fujiyama, and Professor Sakai will participate in the Zero Meat project. Research and Development Task 4 addresses the construction of sustainable socio-economic system through an ethical consumption society based on the concept of watch over food. This project is led by Professor Otsuki, a leading expert in Asian development economies, Dr. Michida of JETRO, and Dr. Honda of Kumamoto Prefectural University, who will quantify the effect of the introduction of the cutting-edge technology developed in Task 1, 2, 3 on society as a whole in a multidimensional manner using an approach that integrated the humanity and science. In addition, we will quantitatively understand the behavior change of producers and consumers due to the introduction of anti-fake technology in the food supply chain. Furthermore, we will examine the prospects for ethical consumption and provide guidelines for building a sustainable socio-economic system. Since this task is an important issue that can be regarded as a compass for all targets, Fukusaki, who is a project leader, and Honda and Muranaka, the leader of task 3 and 5, will also participate in this task. Task 5 is to develop human resources for food solutions and create opportunity through an interactive international program based on the concept of learn food, led by Honda, who has a great experience in international exchange in Asia, driven by biotechnology research. This task will establish an interactive international education program for students and adults from Japan and Asia to study together with diverse members such as Kimura from the School of Human Science, Sugawara and Shiraishi from School of Foreign Study, and Sumimura from the Global Innovation Organization, Sastia from Indonesia, and Ida from the company. In addition, we will develop human resources who can look at various factors related to food, such as economic history, religion, and politics, and look to the future. We will produce human resources who can provide new food solutions that promote social transformation toward low food loss and ethical consumption. In order to achieve the vision of this center, we will cuddle food, love food, bring up food, watch over food, and learn food. Osaka University will make a pioneering effort to realize this global vision and create a society in which all stakeholders, for example, agriculture producer, storage distributor, retailer, and consumer feel happy and environmentally friendly and connected through the food cycle across national borders. So to facilitate this project successfully, Indonesian friends, I strongly want Indonesian friends help us. Okay, thank you very much for kind attention.
Yes, thank you, Professor Fukusaki, for the very nice and informative and comprehensive presentations. We will discuss more at the end of our sessions. Ladies and gentlemen, we will continue to the next presenter. I'd like to invite the second presenters, our presenters today. Please welcome Dr. Sastia Pramaputri. Dr. Sastia Pramaputri. Hello, Doctor.
Hello, good morning, everyone.
Oh, good morning. Okay. I think I would like to say hello in bahasa. Selamat pagi, Mbak Sastria.
Selamat pagi, Pak Andi.
Posisi di Jepang ini ya?
Ya, di Osaka.
Oh, di Osaka. Okay. Ladies and gentlemen, Professor Dr. Sastia Pramaputri is an associate professor in Osaka University, Japan. Maybe please host to show her profile. I'm sorry. Let me introduce first to the audience related to the Dr. Sastia. So, ladies and gentlemen, Dr. Sastia will present the topic. Is very nice topic actually, metabolomic approach to extend the shelf life of tropical fruits. So this is the profile of Dr. Sastia. Her expertise is metabolomics and natural products, and she finished graduate school PhD in the Graduate School of Engineering, Osaka University in 2010. And he has several experience during her career. Research trainee, International Center for Biotechnology, Osaka University, postdoctoral researcher in Osaka University as well. And then she also already published in the very high impact factor publication journal related to the metabolomic topic and etc. So there is no questions about her expertise, and please welcome Dr. Sastia to our event. This is our pleasure for having you here, and the floor is yours to deliver your presentation.
Yeah,
Yes,
thank you very much, Dr. Andi, for your kind introduction. I'm very happy to be here today. Assalamualaikum warahmatullahi wabarakatuh. And yes, and I'm very happy to be invited as one of the speakers. Ibu Puji, Pak Mulyana, Pak Arif is also here. I see. Hello, Bapak, Ibu, and I'm very happy also to share this event with my mentor professor, Professor Fukusaki, who has already made a presentation earlier. So as has already been mentioned by Fukusaki Sensei, we are working on many different kind of research topic related to the profiling of metabolites in various kinds of tropical foods, actually. But today our topic is going to be focused on the tropical fruits, and Professor Fukusaki has already kindly introduced and give us a broad overview of what we aim to do in the COI Next project. And I would like to first give several examples of the work that has been done in Osaka University leading to this point, and what we hope to do during the COI Next project in collaboration with BRIN and also other institutions, our existing collaborators, for example, like IPB University. We also have collaboration with ITB and Indonesian Cocoa Research Institute and so forth. So Fukusaki Sensei has mentioned about metabolomics, and he is our mentor professor, one of the first scientists to establish many different kind of application area in the field of food science. And actually, metabolomics itself is an interdisciplinary research that combines bioscience, because we use biomaterials, and analytical chemistry, because what we want to measure are metabolites. And so we need the knowledge of chemistry in order for us to extract these metabolites from the biomaterials that we are going to use for our study. And we are going to use many different kind of analytical instruments, and therefore we need the knowledge of analytical chemistry. And in order for us to visualize the data in a clear manner, in order for us to be able to understand and digest what happens in the metabolism of the biomaterials that we study, we use the help of informatics. And this process of data mining helps us to visualize the research result in a very clear manner, easy to understand, in order for us to give feedback back to the bioscience. So in our laboratory, especially in my team, I have already applied metabolomics to many different kind of tropical fruits. In the past, Fukusaki Sensei established the very first examples of metabolomics for many different kinds of agricultural products from Japan. For example, he applied it for watermelon, herbal medicine, green tea. And when I joined his team, I tried to also find my own niche by applying metabolomics for something that is unique to Southeast Asia. In particular, because I come from Indonesia, I would like to use this metabolomics technology for things that are relevant for our country, for Indonesia. So the motivation why I started the fruit metabolomics study, especially Indonesian fruits, is because I see that so many very delicious fruits I know from Indonesia is very superior quality, but why it is not exported to Japan? Why there are so many different kinds of fruits, similar fruits in the supermarket in Japan, but it's not from Indonesia? So something definitely is quite wrong, because I know that we don't have problems in production. We also don't have problems with diversity. We also don't have problems with the food quality. I know it's very good. But then what happens? Why it is not available for foreign markets to taste our best quality fruits? And once I identify the reasons, a lot of them comes from the inappropriate post-harvest strategies. A lot of them are already spoiled even before it reached domestic market. So we still have a lot of homework to apply many different kind of technologies combined to improve our supply chain, our distribution chain, and the way we do the post-harvest treatment. And in the fruit study, we have different kinds of application of metabolomics. In the past, we have already used metabolomics to, for example, investigate the characteristic of different pineapple or different banana coming from different cultivars of different regions, and find the unique characteristic of each cultivar or different region. And also we correlate the sensory evaluation and the physicochemical evaluation of fruits with the metabolite profile in order for us to understand if the food has this particular sensory profile, what is the corresponding metabolite responsible for that sensory profile or sensory attribute. And also, most importantly, related to food loss or food waste, then we know also that a lot of problems come from fruit being very perishable. So it has very short shelf life. And before we could understand how to optimize the post-harvest treatment, our laboratory conducted basic study to investigate what happens actually during the ripening. So when fruit ripens, we know that there are many different kind of biological, biochemical changes, physiological changes. For example, from raw fruit to ripened fruit, it changes color, it changes the peel hardness. For example, then once it's ripened, we can open the peel. It changes, it becomes sweeter, less bitter, and so there are many like off flavor that is available in raw food. Once it's ripened, it becomes delicious and it has good aroma. So these are the things that happen during ripening stages. We know the characteristic, but what are the corresponding metabolites that increase or decrease during ripening. This is something that also we investigate in mangosteen, in banana, in pineapple so far. And we also know that, for example, fruits that have been successfully exported to other countries, including Japan, for example, like pineapple and banana, we know that there are some post-harvest strategies that already work domestically. We collaborate with Dr. Feni, for example, from ITB, by which she applied, for example, bamboo storage chamber or chitosan coating in order to prolong the shelf life of banana. But we do not know, we know only that it can delay ripening. We know that it could improve the shelf life, but we don't know how. We don't know what happens. We don't know what changes in the metabolite profile of these fruits after being subjected to this kind of treatment. So this is also something that we investigate using metabolomics to apply these different kind of post-harvest strategies to different fruits, and then investigate what happens in the metabolite profile, what changes in the metabolome after subjecting these different treatments to our fruits. So this is one of the examples that I would like to explain briefly in mangosteen. We know from the literature, there are many different ways that we could prolong the shelf life of mangosteen based on the literature. But what worked for us in here is just low temperature, for example. Once we apply mangosteen for low temperature, we could see that the peel color is hold, for example, similar to day 2, whereas the other reports that have mentioned the different kind of, for example, applying stress inducer like methyl jasmonate or salicylic acid, in our case, it didn't work. Then we investigate, when we compare low temperature and room temperature, what happens? What are the differences? So here we could see that low temperature delays mangosteen ripening process in part. And in the normal case, when we observe what happens during the ripening in the normal case, we see that there is an increase of fructose, glucuronate, galactose, galactorenic acid. These are the ones that are responsible for sweetness. They're responsible for peel hardening. It becomes softer, but it doesn't increase when we subject these fruits in low temperature. So that means there are some, this low temperature is effective to delay the ripening, by which these metabolites that are indicated to be involved in ripening did not show any metabolite changes after treatment. We also could see clearly what happens when we delay the fruit ripening. There are metabolites that increase in normal ripening, and there are metabolites that couldn't increase because some maybe enzymatic activity is halted under low temperature. So for banana, it's similar. So we know that chitosan treatment and low temperature are two of the effective post-harvest strategies that are commonly used in Indonesia now. Chitosan is not as common, but many people are trying to use this also because it's relatively low cost if we produce the chitosan from waste. And then we compare, because both methods are effective to delay ripening. We want to know whether they work under the same mechanism, whether when the fruit is delayed, the ripening, is it the same when we apply low temperature and when we apply chitosan? It turns out it works in a different manner. So different mechanism of banana delayed ripening during storage was observed. So different metabolites were affected by each treatment. In short, we already published these papers, and this is open access. So if you're interested later on, you could Google it. And the low temperature affecting metabolite is related to hypoxia, the low temperature one, while the chitosan affecting metabolites after treatment is related with ethylene biosynthesis. So both are effective. In the phenotype, it looks the same, but mechanistically, it's different. So by understanding these things, we could later on more strategically and more objectively devise appropriate strategies for post-harvest. And in pineapple, for example, we know that we have very delicious pineapple in Indonesia, C4 stage, but C4 stage pineapple lasts only 1 week. before it deteriorates, before it becomes bad. But C1 or C2 pineapple, although it's not as sweet and it's still quite sour, they are mostly the ones that are exported to outside, because under the marine distribution, under sea distribution, it takes about 3 weeks using container from Indonesia to other countries like Japan. And these fruits in the early ripening stage, C1 or C2, can last longer. But then how can we make the C4 pineapple, which has better value, better sensory property, it's more acceptable to the consumer? How can we make this C4 pineapple last longer? So we also, similarly with mangosteen, but mangosteen is a climateric fruit and this is non-climateric fruit. We also compare and we found that upon ripening, when we see what happens during ripening, melesitos was increased as ripening progresses, and inositol is decreased when ripening progresses. And we use this melesitos to coat the pineapple, and we would like to see in the cut pineapple whether melesitos coated pineapple is a better dip. We dip the cut fruits of pineapple into melesitos and see if it could have an effect. And we also add chitosan for enhancement effect, and it was able to increase the shelf life of cut fruits in pineapple. So this is one of the example how we investigate what kind of endogenous metabolites increase during the ripening stage, and then we employ post-harvest strategy by adding these endogenous metabolites, by adding it exogenously, and then introduce this as one of the post-harvest strategy, and we were able to further prolong the shelf life of cut pineapple. So the melesitos added with chitosan was able to preserve metabolite related with antioxidant and cell wall metabolism. And now the latest research that we are doing is using coconut, and we would like to see, because coconut is highly perishable, we know that mostly we use sodium metabisulfate to preserve the coconut, because it can also act like as a preservative, and also refrigerated condition, both in combination is usually used now for post-harvest strategy during the distribution of coconut.
Udah masuk.
Yes. Okay. And we would like to also investigate, this is an ongoing research topic for coconut, very new in our team. We would like to see whether this sodium metabisulfate affects also the quality of the coconut water and flesh, because we employ it, we dip it outside, but we want to know if this that it has an effect also in the fruit flesh itself. And we saw that sodium metabisulfate was able to preserve young coconut, and we would like to see whether there is an effect also in the metabolite profile after subjecting this post-harvest treatment. And this is an existing research topic on mango that we have already collaborated with BRIN, and we just recently published this research in 2021. It was just several months ago, by which we investigate the different characteristics of mangoes in Indonesia. And we hear from this research, we have identified some cultivars of mango have high sensory score, and some cultivars of mango have low sensory score. And in particular, we found something quite interesting, by which we see that Indonesian mango, especially Arung Manis and Gedong Gincu, are high in vitamin B3, and that it has similar export quality with Miyazaki mango, which is the world's most expensive mango from Japan, and as well as Thailand mango that are very commonly traded worldwide as a superior mango. So by this kind of investigation, we could identify what is unique, what is good, what is added value in the diverse fruits that we have in Indonesia. From this analysis, we go on further to then think about, so what happens for the mango that have low sensory score? We know that, for example, Cengkir Indramayu and Lalijiwo have low sensory score in this research because it has high organic acid, so it's quite sour. But we would like to also utilize these fruits because it has high production in order to minimize food waste. So this is an ongoing collaboration with BRIN that is we would like to promote further. Then we would like to use this less preferred fruit, for example, like cengkeh and lalijiwo, and convert it into different kind of product. So perhaps this derivative product later can improve palatability and can be sold, can be used as a source for derived derivative product. Then we could reduce the food waste for less preferred fruits with high production. So finally, this is the end of my talk. I would like to just make a brief introduction about the other thing. This is just a concept. We are still in the very preliminary stage of this research, but we also try to develop a different kind of approach compared to what we used before under the COI Next project. So we know that there are many different kinds of problems of food loss. Fukusaki Sensei has already mentioned, and the food loss can happen in different stages in the supply chain. And mainly, we also can see that, for example, infection by microbial contamination is one of the cause of food loss because the food becomes spoiled. And during transportation period, high perishable foods such as fruits and vegetables can easily be spoiled following the senescence. So we use the banana, especially Cavendish banana is a very commonly traded fruit. Even we have many different kinds of banana in Indonesia. We use Cavendish as a model banana. And from the banana plantation, from harvest, packaging, transportation, the key point also here in refrigerated cargo, and the ripening chamber. This is how the supply chain happens for banana, especially when it's exported to other country. And we would like to investigate, for example, identify problems in the banana supply chain, such as here, maybe it happens bruises, banana disease, for example, cannot ripen. So many different kinds of problems will result in food waste. So in order for us to identify what are the common problems, by communication, by intensive discussion with Indonesian friends, then we hope to identify the biggest problem in the supply chain of fruits, and later on work together to devise a good experimental research design in order for us to tackle these problems in the present supply chain. So I have already mentioned before many different kind of examples how metabolomics can be used to investigate the effect of post-harvest and to study ripening and so forth. But it has not been applied in the real life setting, I mean in the industry. So we hope under this COI Next, we could collaborate with Indonesian friends from the early banana plantation, from harvest until it arrives in Japan, to work on the different parts of the supply chain, identify the problems together, and then once we identify the problems, we could exchange ideas on what kind of approach is suitable to tackle and solve the issues of each problem in order for us to get the maximum output, maximum outcome of this research under the COI Next project. So one of the main ideas that we have in mind right now is to combine existing technology with metabolomics approach. For example, we have a good expertise in metabolomics, so we would like to identify volatile components that mark the onset of spoilage, early ripening, and fungi infection, for example. And later on, we would like to develop a sensor to detect early spoilage before the spoilage happens. And we can prevent loss before shipment, or we can also prevent loss during storage and distribution. So one of the ideas that we have in mind is later on we would like to build a gas sensor detection system to monitor the quality of fruits during shipment or storage based on the volatile component marker that is suggested from this study. So this is one of the research ideas we have in mind right now. But of course, we are very open to having very good discussion in this meeting and hear the proposed topics from Indonesian collaborators from BRIN that will be speaking also today. and later on work together to solve this food loss, in particular for tropical fruits in Indonesia. That is all for my talk. Thank you very much for your kind attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much for your excellent presentation, Dr. Sastia, and also as well as the what can I say, the main idea for collaborating project, upcoming collaborating project, and we will discuss more in detail at the end of our sessions. Okay, ladies and gentlemen, we had already 2 presenters from the Japanese friend, from our Japanese friends, and then now we are going to listen and see how is going on in our Indonesian friend, and from our Indonesian friend will be driven by Ibu Puji Lestari PhD and Dr. Mulyana Hadipernata. Ibu Puji Lestari PhD is the chair lady of our research organization for Agriculture and Food BRIN Indonesia. Hello, Ibu Puji.
Hello.
Good morning.
Good morning.
Yeah, sehat Ibu. Yeah.
Alhamdulillah,
thank you so much for attending this event and discuss with us. And Ibu Puji Lestari will be delivered the topic of overview of research and on reduction of food loss on selected tropical food in Indonesia. And here we see the show the Ibu Puji Lestari profile. She is the chair lady in our research organization, and her area of expertise is molecular biology. She finished her PhD in plant biotechnology, Seoul National University in 2005 from South Korea. She already has a lot of experience in her career as a researcher. Now is the senior researcher of Indonesian Center for Agricultural Biotechnology and Genetic Resources, Research and Development, and researcher of Indonesian Center for Rice Research. She is as chair lady of research organization for food and agriculture in National Research and Innovation Agency BRIN. And in last decade, she also already published in the high impact factor publication journal, and working related to the isolation of microbes producing enzymes, amylase, silanase, etc. Okay, we are going to invite Ibu Puji Lestari to present her material. Ibu Puji, the floor is yours now. Please, Ibu Puji, 15 minutes more or less. Thank you.
Thank you, Mr. Andi. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi Bapak Ibu semua. Good morning everybody. First I would like to thank to Professor Fukusaki and Dr. Sastia for delivering very great talks, and that can be useful and beneficial for our collaboration between BRIN and Osaka. Could anyone help me to share screen of that PowerPoint? Okay. Actually, I'm representing our RO, Research Organization for Agriculture and Food. Here I would like to talk about, is that the title? Could you get back to the first slide, please? Reduction of food loss on tropical fruits. Next. This is really just, we would like to share this information so we can later, we can have, a better match or link between a research program in BRIN and what we want in Indonesia, and then what kind of technologies we already proposed or have by Osaka University. So here actually, this RO, yes, we focus on the agriculture and food. So considering the challenging of agricultural and food loss in Indonesia, actually is the decrease in the quality and quantity of agricultural and food product in the supply chain can include in the production, cost, harvest, storage, as well as processing, which already mentioned also by FAO, and especially for the magnitude of food loss and waste in Indonesia from 2000 and 2019 reached is very high, usually 2348 million, and have the impact of economic loss. Next. And that's why it's very important to reduce food loss and waste. We have to undermine the sustainability of our food system. When the food is lost or wasted, all the resources will go to waste. In addition to this, can contribute to greenhouse gas emission and finally also contribute to climate change. This food loss and waste also can have negative impact on food security and food availability. Next. In addition to that, so this issue, especially food loss, of course, because Indonesia, we have a high population. So the increasing population actually has a dramatic impact on this quantity and quality of food demand. And recently also the focus on long-term or even mid-term of or goal of this BRIN, especially this RO of agriculture and food, we try to cope climate change. So this food and agricultural system will also continue to be challenged. So this way, adaptive kind of varieties or technology is very important to respond to this threat. And the complexity of agricultural products, supply chain, and limited technological support also can supply the product stock that especially can threaten food security. So focusing on these horticultural crops, because fruit is belong to this crop. So this crop is very dynamic. So it's a lot of those about fruit are mentioned by Dr. Sastia, also the Professor Fukusaki. So in Indonesia, so we concern how our horticultural crop can be compete in the global market. So this is why it's demand for both quality and price competitive products. So we have to solve this problem. Next. This one, in addition to that, so because we have diverse genetic resources, so on the basis of livestock or other like forage crops in Indonesia, we try to explore the potential of black throated fly. This is example, because in Indonesia we have a lot of waste from oil palm. So here actually also other like fruit. So we try to explore the potential of this maggot also or the SF here in the recycling many form of waste in the environment actually here. So with the benefit of the high content of protein and also a lot of food waste in the single or combination with an animal manure is effective to maggot mass production. We also try in this next, research that maggot also can be a part of this research on how to reduce the food loss. Next. Here is a lot of direct and indirect factors that can drive this agricultural food loss and waste. For example, lack of implementation of good handling practice, poor storage quality or quantity, and lack of information of producer and consumer, and etc. Here is including the direct one like technology limitation, poor harvesting technique, poor of packaging. So how we can reduce the food loss in Indonesia? Not only the harvesting, post-harvest handling, post-harvesting process, but also industrial processing, packaging, storage, also selling. This way, application of technology and standard present procedure are very important. Next. So to try to solve all of the problem, so in this RO of agriculture and food, we have 6 research centers. Here, I would like to introduce briefly the core research of each research center. So how each research center can contribute to solve this problem, and collaboration not only with Osaka, but also in BRIN, we have other research organizations also can work together in this reduction of food loss. First is the Center for Food Technology and Processing. There are 5 core research. First, material is to focus on the post-harvest processing on food material and also natural material, especially from Indonesia for functional food, and fortification and biofortification. And on the basic process, we also work on the develop technology processing for plant food product and technology processing for animal, also for marine. So in the advanced process, we focus on the development of beverage product, functional food biotechnology, and also in microbiology. So in the preservation, innovation of food safety detection and food preservation technology and food packaging technology also are very important here. And here in Indonesia, since most of Indonesian are Muslims, so here for food program, we also have a priority on the development of halal research and advanced with technology here, and revitalization of food security, especially for stunting mitigation. Next. And so the strategy program for until 2024, because in Indonesia we have now we already have the term of reference or proposal that should be submitted to national level in Indonesia through BRIN. So in this research center, the strategy program focusing on the stunting handling. So as we mentioned that food fortification and biofortification is a both sides. This biofortification is really mostly for genetic material, so also food fortification and the exploration of local material. In Indonesia, we have a lot of local genetic resources here, so we have to also optimize their potency, and diversification of food and beverage is very important in Indonesia. So for processed food packaging, here commercial sterile packaging and preservation with new technology also for the packaging, especially for edible, active, small, and degradable packages, also one of our goal. And in the halal research and innovation, we have a homework. Really, it's very big homework with how we have a detection. I mean, fast and easy detection on this halal and halal food or product. And also we try to explore the substitution of halal assurance. Also from not only from marine as well. So here it's for research. We try to get or find a lot of material or substitution and exploration more in this BRIN, especially for this technology and the process of food. Next. And for this, a lot of research group here, so just examples, a lot of research group in this research center support this research activity. Next. This example, innovation product is innovation of chocolate, and it hopefully can attract new tourists, and it's actually because the packaging is also contribute to the how to we reduce the food loss. So in BRIN we already have some this technology, and it's already also go not only to small or micro or medium enterprises, but we try to go to industry. Next. For the traditional kind food technologies can be benefit to protect food from microbial, physical, and chemical spilling problem, also photochemical reaction, and also the gas permeability spillage problem and radioactive. This technology is really now distributed across Indonesia. You can see the map here that this traditional food for canned food development. This is contribution of this research center. Next. And here's this ongoing research for processed food and could be potential for fresh food as well. We use indirect plasma like ozone treatment, and direct plasma is we use cold plasma treatment and also gamma irradiation, because in this RO we have maybe almost 1,000, I think. Soon the human resources in RO will, totally will join in this RO. So we have a lot of expertise, and scientists can contribute on this research. So here also we try to get how this activity, this research can be beneficial for this food loss with Osaka. I think. Next. So second one is the Center for Agri-Industry. This core research is focused on post-harvest of agri-industry product and extraction and processing of agricultural product, livestock, fishery, and marine, and also downstream agri-industry of estate crops. And the strategic target is really not only for utilization of natural resources in sustainable agriculture. Here this can provide added value for agri-product, but also the development of the agri-industry through processing, plantation, agriculture, animal husbandry, marine, and also forestry product. Next. And the third one is Research Center for Appropriate Technology. The core research here, this research center also supply other technology. The research supply to other research center and other research organization. And this research center focus on and have mandate for developing appropriate technology, equipment, and process for pre and post harvest, and equipment and process for local agricultural product. Also food storage, packaging, distribution of agricultural product, utility research, system integration, and commercialization of appropriate agricultural technology, as well as how to manage this agricultural waste and agricultural food of UMKM. Is like, yes, micro, small, and medium enterprises. Next. This is this research center. It's mentioned, I think, by the previous speaker. But mango is one. Yeah, mango is one of the fruit that most everyone might like mango, and we have diverse varieties of this mango, especially local. Not only local, some of them are maybe introduced from other countries, and this we have experience of losses of mango fruit during post-harvest handling are caused by harvesting because improper maturity stage, poor post-harvest pre-treatment, improper packaging, inappropriate storage temperature, also distribution condition. Next. So for this, actually, this research center have experience to carry out this post-harvest pre-treatment on mango. This post-harvest pre-treatment are suitable for fruits with the non-chemical pre-treatment like hot air treatment, hot water treatment, or irradiation, also chemical pre-treatment. Next. Using hot water treatment is some studies also demonstrated that the benefit of and the potential of this approach, and compared to the result from this research center, is with specific temperature is effective, or with the specific period, like 30 minutes, can eradicate eggs of fruit flies and to maintain the fruit quality. And with the hot water treatment also is highly recommended practice at farmers due to its cost effectiveness and simplicity. Next. So in addition to that, hot water treatment also control atmosphere storage. So hopefully we also hope that can use this technology. Next is a method. So with the appropriate technology. So next is this is about our other research center, horticulture and estate crops. The fruit is belong to horticulture. So it's one of our core in the research center as research organization. That the core research basically is on the genetic resource management here, the bioprospecting. starting for exploration, integration, everything, also the conservation, screening, using not only the conventional, but also we use the advanced technology. And plant breeding here, because it's mandate of this RO, we have to release or develop our genetic improvement of any varieties to get a better new varieties to be released. So this in the breeding, we use conventional and biotechnology breeding, like in vitro culture, mutation breeding, etc. So here, actually, from the genetics like metabolomics. So that's why we need assistance from Osaka University. And the smart agriculture technology development for present intensive and adaptive agriculture by using IoT and agri-industry. Here also is one part of this research in this research center, and productivity and quality improvement, and also innovation technology for specific location. Is the main scope of research activity in this research center. Next. It's just example for mango. So because hopefully it can be a marketable this mango, but a lot of problem like slight anthracnose infection. If we could have, varieties resistant to this disease or improve on cultivation system, and also have a good management of pests and disease, hopefully can reduce this food loss on this commodity. Next. This way with metabolomics, maybe as tested by this from Japan. So a lot of homework and a lot of that goal. We want to increase how our fruit, tropical fruit, with several activity and this kind of output from the genetic resources and from how the metabolism knowledge on characteristics of defense frame or biomarker actually link it to any specific trait or important trait for this fruit. Those can be beneficial for us. Next. So as an example, we have done several research, not only on phenotypic, but also on the molecular biology approach. We did with the universal SSR markers, and also we have a plenty and diverse of Indonesian mango based on the fruits, fruit peel, leaves, wood, and bark of tree. In Bahasa we call Queni, Hipakel, Kasturi. This is our local name, but this really is very, very amazing, and that we have this kind of rich diversity. So for this, what the prospect metabolic study is like development of biomarkers to track the ripening stages could be very useful. Next. In addition to that, it is coffee from estate crop. So we have diverse of from coffee Arabica, like Robusta, and Indonesia also consumer of coffee. So we also have the technique for in vitro conservation, especially for potential extraction, because this technique is some of them are genotypic dependent. So with the metabolic study, hopefully this metabolic class determination affected by coffee breeding can be determined with different metabolic approach. So also the coffee metabolism can aid future attempt to yield the coffee seed or with the desired trait, especially like a best flavor type or others. Next. And is this animal husbandry, this research center. The core research focus on the genetic resources management, like exploration and characterization, the same as horticultural research center, and also breeding and nutrition technology for livestock. They support the efficiency of livestock production and technology for physiology and reproductive to increase livestock productivity, and also farming technology. Also, we try to carry out on the information about the information technology for livestock genetic database that can support this information is very, very important to provide this kind of information, especially in Indonesia, that can support other research activity. Next. As the last, it says food crops, research for food crops. Because it's a staple food of Indonesia, rice, and then also maize and other cereal, legume, and tuber crops. And this core research and this research center is principally is the genetic improvement or development new variety, using not only conventional but also technology. Here also we have to do seed production even with collaboration with the company, but not BRIN, because BRIN just do research. Also integrated crop management technology is we need to do like agronomy, physiology, and also under interaction among nutrient and others, and also how to control and manage the pests and disease and development of this new variety. Not specific location, also for general in Indonesia, that depends also how we answer or how we for is that to face the hot issue or current issue, because Indonesia is now it's only okay for national program. Usually we have already grand design for what is the commodity like rice or chili that can impact for this economic aspect, but also how BRIN can answer every current or hot issue that we have to do any research to answer or this kind of challenge. Next. Okay, this is future research. I'm sorry, I apologize, because this is actually a lot of idea and topics have here in our mind. So how then we can arrange with like a big project. But here it's from the five, because we focus on fruit. So from the 5 research center, we try to accommodate this all of the idea and proposed topics to be researched from the agro-industry. This is just the idea, but how we collaborate among this research center and with other research organizations. First, on the agro-industry, this focus on the agricultural fresh product handling, also system and industrial management. And this for agricultural fresh product is improving the quality and added value of agricultural fresh product and increase shelf life of agricultural product. Also for reduction of loss on agricultural product. In agro industry, this focus on, as we mentioned, is not the food crop, but others, including fruit. And the system and agri-industrial management, how we can have the agri-industrial production system efficiency. And to support this, also from appropriate technology, as an example, how we could develop equipment to control atmosphere storage with cold plasma and ozone technology. Also how to combine hot water treatment and control atmosphere storage to extend shelf life of fresh mango fruits. Or maybe we can also apply to other fruit. Next. And from the technology and processing of food, also a lot of idea actually that from the like high nutrient food, we need also alternative protein, analog rice, analog meat, and for nutraceutical natural ingredient exploration is very important, that how this protein can identify, also how to develop this probiotic, and also how to preservative. And then the food additive here, we can have this kind of fruit based on the how to increase the flavor, aroma, color, or preservation out of the technology. And others for food preservation, at least there's technology can maybe more than 6 months. So in addition to that, fast and easy food detection, this halal research and how to detect from the contaminant and adulteration, also food packaging innovation. Next. So the say for suggested research in this collaboration is possible that we could carry out together. We can make priority, but for this food analog or bioengineering, preservation, advanced food processing technology, how we can do this. Next. As for metabolic condition of Indonesian mango, how we have biomarkers that can be useful to identify the nutritional and phytochemical compound of Indonesian mango, germplasm, and also to track the ripeness stage to support effective storage method. This the outcome could be not only for the utilization of the mango for the Indonesian mango to mitigate global food insecurity, but also how this food ripening process could be used to develop effective storage strategies and reducing post-harvest losses. Next is from the not only from plant food, but it's from, it's mentioned, palm oil in Indonesia. Actually, yeah, we is one of the first producer in the world. And we have a lot of abundant organic waste in Indonesia, and then how we could use this palm oil waste for the mass production of maggot to support sustainable livestock feed. And several research activities are here. It's just we need this kind of research activity that can be worked together with Osaka. As an example, this is how to the nutrient of BSF, and then residue of as material and application for biofertilizer, and also how from the economic analysis, this also mentioned from the Fukuyama that some economic also you work with that aspect, so how we can work together. Next. I think this is all of our presentation from RO for agriculture and food. Thank you very much. Yeah, Mas Andi, thank you.
Yes, thank you so much, Ibu Puji Lestari, for the very comprehensive presentation related to the our institute in BRIN, especially in Research Organization of Agriculture and Food, and the research institution inside this organization, as well as their work. So in this case, I didn't stop Ibu Puji Lestari presentation, because I think this is a good moment for our Japanese friend to know about our institution and our works from now on. So we will have idea how to link each other between our side, Indonesian friend, and Japanese friend, not limited to the fruit in this case, but also other sector, agricultural product in general. Okay, ladies and gentlemen, we are now going to the last presentation, but not least, of course, the last presenter will be Dr. Mulyana Hadi Pernata. He is the head of Research Center for Agro-Industry, BRIN, Indonesia. Let's say hello to Dr. Mulyana. Hello, Doctor. Selamat pagi. Good morning.
Selamat pagi. Good morning, Dr. Andi.
Ya, sehat.
Nice.
Ya.
Alhamdulillah.
Ya. Alhamdulillah. Yeah, ladies and gentlemen, Dr. Mulyana Hadi Pernata finished his PhD from Food Science, Ehime University, Japan, 2016, and his expertise is related to post-harvest technology in general, and he has already a lot of experience related to the last his career as the researcher of Indonesian Center for Agriculture Post-Harvest Research and Development, now converted to BRIN. And then now he is the head of Research Center for Agro-Industry Research Organization of Food and Agriculture, BRIN. And he already published in several journals, and his work we can see here undeniable about his expertise. So yeah, we will give the floor to Dr. Mulyana, but before I give the time to Dr. Mulyana, I also would like to say to the audience, so please feel free to type your questions here that we will resume, and we will make a session for question and answer after Dr. Mulyana presentation. So Dr. Mulyana, please deliver your presentation. Floor is yours now. Thank you. Hopefully not more than 15 minutes.
Okay, maybe I more fast.
Yeah, okay, thank you.
Thank you very much, Mr. Dr. Andi. Let me share my material. Okay, you can see. Mr. Andri?
Yeah, it's very clear.
Okay,
please.
thank you. Okay, first of all, I would like to say thank you for this meeting and the Honorable for Chairman of Research Organization for Agriculture and Food, Ibu Dr. Puji Lestari, and then Principal Investigator COI Next Project Osaka University, Fukusaki Sensei. Nice to meet you, Sensei. And also the Honorable Prof. Sastia Prama Putri from Osaka University. This is the second chance I meet you again, Professor Sastia. Nice to meet you again. And also Head of Research Center for Appropriate Technology, Head of Research Center for Food Technology and Processing, Head of Research Center for Horticulture and Plantation, Center for Food Crop, and also for Animal Husbandry Research Center. And for all seminar participants, professor, senior researcher, also research colleagues, and thank you for attending this seminar session. Okay, Assalamualaikum warahmatullahi wabarakatuh. I would like to share about our target in Research Center for Agro Industry. This is utilization of natural resources in sustainable agriculture by providing added value for agro products. And also we want to develop the agricultural industry that can reduce agricultural losses, also maintain quality and increase added value. And then we have the scope that's here's agricultural fresh product handling, and then intermediate product processing for agricultural industry, also reduction on loss of agricultural product, and then the four is processing of downstream product and derivative, and also agricultural byproduct processing and valorization, and increase the added value of agricultural industry product for export purpose and production system and buy system. And here's our future research direction. Here's we have the four program. For first is agricultural fresh product handling, and then sub program is here you can see that improving the quality and added value, and also increase shelf life of agricultural product, and then reduction of losses of agricultural product and post-harvest physiology. And for enhanced competitiveness in agro industry, we also have program for intermediate product, and we support from a sub program extraction process and product development. Also sub-program purification process and product development, and product development based on chemical, physical modification, formulation, and synthesis. And third program is downstream product. And you can see here we make some product development, and also product has the high competitiveness. And in the fourth program, of course, technology without system or base system is not complete. So in agro-industrial program, we have the agricultural industrial biosystem and program in agro-industrial eco-product handling management. And before going to the main topic, but here's the characterization or about the mango. I would like to share about the one example of research conducted by Research Center of Agro-Industry. This is related the coating from palm oil as a basic material, and coating application are used on a fruit and can increase their shelf life. So this is very useful for us, or for a farmer, or for maybe a company, if we can maintain the freshness of fruit. In this case is a mango. We can make increase the financial income for the farmer. So usually the farmer using the beeswax or as the coating, and then we make some new technology from coating from derivative from oil, palm oil. So we can we can make this solution is more cheap than the established wax in the world. And also in here in hello, also our product is our technology is edible coating category. And why I saw this our research in agro industry, I would like to present to all audience that's this is important. The technology is very important to reduce reduction of the food loss in fruit industry. And our research has the support from Agency for Oil Palm Plantation Fund Management, or BPD PKS. Okay, and then another here's our recent. I would like to now talk about the recent research collaboration between Research Center for Agro-Industry and Department of Biotechnology, Osaka University. And here's that for first project is characterization of 5 Indonesian mango using gas chromatography mass or GCMS based metabolic profiling and sensory evaluation. We have some duty in our side in Agro-Industrial Research Center is some kind like a sample preparation, and then physicochemical evaluation, and also sensory evaluation. And in the side of the Osaka University, they has a duty for metabolic profiling and data analysis. This is maybe I fast in here. This is some like data for physicochemical property. We can say total soluble solid, and then like the texture or total titratable acid. So we can compare from many variety or of the mango. Chokanan, Miyazaki is like a mango from Japan, and we have this is 5 variety of mango: Podang, Gedong Gincu, Arumanis, and Cengkir. And then we can compare with our Indonesian fruit with another fruit from another foreign country. And Also, we do the sensory analysis. We can see from sweet aftertaste, and then bitter aftertaste, aroma sensation, juicy, sour, fruity, and another sensory analysis. And then we have the data from this, and also we use the GCMS analysis to find out the metabolic in mango, and then we find that in our mango is category of the include based on the PCA analysis. We can see that the 47 annotates hydrophilic, and then low molecular weights metabolic, and 48 unknown metabolic detected by GCMS analysis. And this is the second project with Osaka University related the metabolic and sensory profiling of processes in Indonesia mango. So after for the previous study is the fresh mango, and then in the second study is the processing mango. So we make this is in this case is a freeze dry mango, and then dehydrate mango and mango letters, and then we check again with also sensory, and then also the metabolic, and we get the some data. Okay, maybe here's previous Dr. Puji Lestari already explained about research proposal for agro industry. I would like to explain again here that of course the target, the focus target in our research proposal is agricultural fresh product handling, because all of many farmers or many company is need of this technology, and not only technology, but also we care, or we have a target about the system and agro-industrial management. For example, in this case is agro-industrial production system efficiency. We know that supply chain management is very important to reduce the food loss in Indonesian fruit industrial fruit side. If we can simplify the supply chain in fruit trader or fruit market, of course we can reducing our food loss. Okay, for summary, we want to talk about the profile and characteristic of the fruit are the basis data for development of fruit cultivate in Indonesia. For example, by knowing the profile of product, for example, a fruit that has a long shelf life or short shelf life, we can create techniques and method for post-harvest handling and post-harvest processing. In the previous slide, I talked about the how to handling the mango by coating or formula coating. And then, okay, maybe its time is over because Mr. Dr. Sastia has another duty. Okay, thank you very much. This is Professor Sastia when I meet in Jakarta. Okay, thank you very much for listening. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Dr. Mulyana, for the presentations. Yeah, we will have actually we in our room we have information that Dr. Sastia and Professor Fukusaki has another webinar in 11 actually in Indonesian time. So this first this first session, we are going to the question and answer. But before we go to question and answer, I will give opportunity or time to Dr. Sastia to conclude this presentation after listening and see the Indonesian friends presentation, as well as we will give time to Professor Fukusaki. Yeah, please, Dr. Sastia.
Okay, thank you very much. I'm very pleased to see that there are so many ongoing research work in BRIN that I think are very suitable and very much in line with what we are trying to do and what we would like to propose. In particular, in the agro industry, the main focus of today's discussion by Dr. Mulyana mentioned several post-harvest strategies like the coating to replace wax. This is very unique. And also, Ibu Puji mentioned about the use of water heat treatment, because in Japan, we have a problem that Indonesian fruits cannot enter Japan because of the fruit fly problem. So later on, perhaps metabolomics can also work together to check the comparison between, for example, fruits that are using the VHT treatment and the water treatment. And there are many research work that we could identify together that is already presented here. Actually, it's just connecting the dots. And I think very promising collaboration, especially research topics also in other centers that Ibu mentioned, can easily be adopted, because we have already the expertise to analyze many different kinds of food products. And we have already optimized also the sample preparation, the way to prepare the sample and bring it into Japan. So these things we have a very good experience in the past already. So I think by understanding and identifying the many different research proposals that are presented today, later on we could follow up by discussing in a more detailed and technical level on how we are going to implement new research topics for collaboration. So I'm very pleased that we have this sharing session today, and I hope that more research topics can be explored together, not only with our team in metabolomics, but Fukusaki Sensei also mentioned that in our CO&X project, we have different research divisions, for example, working on the economic and also social impact. And also we have the collaboration, for example, using the freeze-dry technology and to use the orphan crops, many different dimensions and the research avenues we could explore together to expand the collaboration and strengthen the existing collaboration and partnership. That is all for me, and I really thank all of the help of the collaborators from Indonesia, especially from BRIN, for hosting this really great sharing session. Thank you very much.
Thank you very much, Dr. Satya. We are appreciate as well. And the next comment will be from, we give session to Professor Fukusaki. Fukusaki Sensei, please give a comment after hearing and listening Indonesian presenter. Yeah, please, Professor.
I really appreciate your beautiful organization, and I really enjoy the beautiful topics. And I once again recognize that different direction would be necessary to enlarge the shelf life. So as Saskia mentioned, in Japan, the importance of the fresh ripened fruit is basically prohibited in principle to prevent fruit fly installation. In the case of banana, for example, only green banana allowed to be imported. Therefore, technology to put green banana to sleep while maintaining their physiological activity, and technology to wake up sleeping banana and ripen them after importance are very much important. So through this principle, in Japan, only banana and avocado and kiwi fruits and several kinds of tropical fruits are imported effectively. So if we develop the very beautiful technology to thrive the physiological activity and safely transporting technology and the good technology to wake up and to facilitate the ripen after importing. So we can import the beautiful fruits from Indonesia. So Indonesian problem is a little bit different. So your problem is development to improve the technology, prolong the shelf life after ripening. So I'm sure that we can provide some technology to facilitate your technological improvement. And we require Indonesian friends corporations help to facilitate our technology. So I'm sure our collaboration surely make a beautiful research fruits and achievement. Thank you very much. So I really expect the next time sharing session. Thank you.
Thank you, Fukusaki Sensei, and we are glad to having if you may be with us until finish, because we have two questions here from Dr. Satyajit and Dr. Marcus. We will give opportunity to Dr. Satyajit first to please ask your question directly to our presenters today. Please, Dr. Satyajit. Please open your mic and then just feel free to directly ask your question.
Thank you, moderator. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
As my background here, I'm from PARD, Badan Litang Pertanian, is now in process of come to BRIN, but the background will be fade and come to the BRIN. So but whether the researcher is come to the BRIN is maybe yes or maybe not. So at the moment we still at the maybe. Okay, I just give some reminder to the situation in Indonesia. Maybe as one of the root researcher or horticulture researcher, first is maybe Bu Putri Indonesia is have so many islands. So center of production is in the one place to another. So maybe need to also to consider what is called the characteristic of transportation from one island to the other island before we export it. Then fruit fly. Fruit fly is a problem in here, and the solution from the market in Japan is vapor heat treatment. It's only vapor heat treatment in there. If we have some because so far Malaysia has one, Thailand has previously, Philippines has also the vapor heat treatment, by Indonesia not yet so far. So if we need an alternative for on the vapor heat treatment, with collaboration between Indonesia and Japan, because price of the mango in Japan is very fantastic. So if we have an alternative for treatment, then we have to also join with quarantine in Japan, maybe also Indonesia. that we saw that we have treatment that they are really powerful to the fruit fly other than paper heat treatment. In here, the maybe in BRIN is not come to the active exporter, but I just so there is industry already exists from the big company like Chem Farm. Chem Farm is based in Semarang and make it a dice and frozen mango. I think mango and pineapple, maybe also melon. The second one is KCP 21. It's based in Lampung. Process of pineapple already established, I think. So that's why profile of export is so far is from the company, and then they now convert from banana to pineapple. I think already started filling the market for global, and some of them is also a local market. And then PT Pura. Sorry.
Pardon me, Dr. Satyajit.
Yeah.
Yeah, I think your question is addressed to Dr. Sastia, but unfortunately, yeah, she has leave because she has another meeting as well. So I think we will compile your questions and we will send to her. Then after this webinar, she will answer to you directly. Yeah.
Okay.
Yeah. Okay. Is there any other question that we'll compile or?
Probably also Dr. Mulyana and Bu Dr. Puji. Yeah. Maybe with the project of metabolome.
But pardon me, Dr. Satyajit, may I give time to Dr. Sastia if she is still here to say goodbye, probably?
Yes, thank you very much. Yeah, we are very sorry. Professor Fukusaki and I have another seminar, our PhD student seminar at 1 p.m. So we would like to compile the questions from the audience today. And as mentioned, every 2 weeks, we will have this setting session. So perhaps in the next setting session in mid-June, we will have a longer time for discussion. Then for today, I really thank everyone and for the questions from Dr. Satyajit. I hope I could follow up the answers to your questions by email or in the next sharing session in the mid-June that will later be announced probably soon. Yes, thank you very much, Pak Andi, Ibu Puji, Pak Mulyana, Pak Arif.
Thank you so much as well.
Fukusaki Sensei, we may be excused first while the discussion will continue here. Okay, thank you very much, everyone.
Thank you very much. Thank you very much, Dr. Sastia. Bye-bye. See you next time. Yeah. Thank you so much, Professor Fukusaki as well. Oh yeah, I'm sorry because I just know that both of you guys have another meeting. So thank you so much for joining us today, and we will wrap it up all questions to you directly. Yeah, bye bye, and we will continue this session. I think Dr. Satyajit may type the questions and we will collect it. And the second one is from Dr. Marcus, probably. Dr. Marcus,
Yeah.
Can you one or two questions? Yeah. Yes, we will collect if to whom you address the question, please.
Yeah. Okay. Can you hear me, Dr. Andy?
Yeah.
Actually, my question to Professor Ichino and Dr. Sastia, but they are left at the moment. So my question is, when we see the presentation from Professor Ichino, he explained about the waste of fruit might up to 33% from the total fruit. So quite huge for the percentage loss compared to intake only for, I think, 67%. Now the problem we have is how do we measure? Is there any method available to measure the loss of fruit to get the overall loss of 33%? Is the method can be applied for every individual fruit? Like the same method, we measure the loss for pineapple, mango, stem, and etc. So I think we need the right method to use to determine the magnitude of individual fruit loss during the process, because I don't think that every individual fruit will lose up to 33%. Probably we had varied, let's say from 20 up to probably 40, but depends on individual fruit. Not can be applied for general rule to say the amount of fruit loss up to 33% at all. That's the first question. And the second question is to Dr. Satria about talking on metabolomic. Now the question is there any information available about the negative impact of the using the technology, because they use the science, bioscience and chemistry? Now the question for the negative impact of human health, let's say probably from cancer or another sort of negative impact on human health. So I will need more clarification about the statement for using the metabolomic in processing to increase the shelf life of the fruit. Dr. Andy, I think that's all. Thank you.
Oh yeah, thank you, Dr. Marcus, for the questions, and we'll write it up into the compilation, and we'll go back to you after we get the answer from both of Professor Fukuzaki as well as Dr. Satya. Okay, ladies and gentlemen, we also get the question actually from the chat room. There are three questions we have already mentioned here, and unfortunately this is also these are also addressed to Dr. Satya and Professor Fukuzaki, but it's okay. I will just read this from Nurul Hidayatun. Is there any possible contradiction between the approach to reducing consumption loss with food safety issues and standards in Codex Alimentarius? And then the second question comes from Nur Alim. A short question for Dr. Sastia. For fruit case, the metabolomics approach is profile the metabolite affecting the ripening process. What about for the food loss of food product affected by microorganisms? Can we use the metabolomic approach to profile the food product that are deteriorated by the spoilage of pathogenic bacteria? Okay, I will deliver this question to Dr. Sastia later. And then the third questions come from Anas Fauzi. Fukusaki Sensei mentioned in his presentation that religion is one of aspects to be considered. Is it related with halalness? Okay. Yeah, thank you so much for the questions from Dr. Nurul Hidayatun, Dr. Nur Aim, and Dr. Anas Fauzi. Again, we will compile this question and deliver to both our presenters. But maybe Ibu Puji Lestari and Dr. Mulyana may have a comment for all of the question from the audience. Yeah, we will give a time 1 minute each probably. Yeah, please, Dr. Puji.
Sorry, Mas Andi.
Yeah.
Yeah. Is there any questions for us? So
actually, there are no questions from for Dr. Puji. Yeah, Ibu. Yeah, Puji for wrap up. Yeah, give a close comment. Yeah. Closing comment.
Thank you very much for everyone. And I think most of participants here are BRIN human resources and researcher are going to join the BRIN, and it will come soon. I mean, in our organization. I mean, in the agency and the BRIN. So actually, this is a good opportunity that we have first sharing session in collaboration with Osaka University, and it's very important for us to start and to initiate this collaboration on to reduce food loss, and especially on fruit, but we can extend to others, and with the 6th research center and the RO of agriculture and food, hopefully it can strengthen and the research capacity, also the capacity building. Because this collaboration is not only for research, but also capacity building with Osaka University. So this today's topic also I think is very good that we it's kind of for brainstorming. I mean, and we can match and with what our wish with the research and what they have, the technology that they already advanced from Osaka. And here a participant in this webinar, and hopefully this webinar can be fruitful to all of us. And please, when you all join to this, maybe some of you join this RO, food, agriculture and food, and you can also contribute for this research activity in collaboration to explore this how to get the output, and of course with your expertise. Yeah, Mas Andi, thank you so much. And
Okay, thank you so much, Ibu Puji Lestari. Okay, ladies and gentlemen, this is the end of our sessions, and then I would like to conclude our discussion and sharing session. First, the X technology consists of several tasks that working together and potentially collaborate with Indonesian part. And the second one, post-harvesting technology play an important role to decrease the food loss by the appropriate monitoring technology, such as metabolomic. And metabolomic approach give the insight related to the how is the strategy of during the post-harvest technology treatment for reducing the risk of food loss. Each fruit has a unique characteristic related to the ripening process, so the strategy will be different, how the ripening process should be treated. And the collaboration project should be carried out to solve those kind of problem we are coping. And then deeper and intensive communication between Indonesian part and as well as Japanese part should be strengthened, and then should be continually to force the target of our goal to decrease the food loss to 18% in 2030. And the last conclusion is issue can also be expanded, not limited to the fruit commodity, but also agricultural product commodity in general, including the marine product and the animal product. Thank you so much for this is for the conclusion of our discussion, and we hope that our collaboration will continue to another deep and in detail discussion. Will be in several tasks related to the link with the Japanese part. Again, before we close the session, I will be give the thanks so much. Thank you for everyone for attending today's webinar with the topic reduction of food loss on tropical fruits finally, let's give the applause for the speaker as well as for you all. Thank you.
Mana kabar, Bu Puji?
Yeah, on behalf of Research Organization for Agriculture and Food, BRIN, thank you for joining us today and have a great rest of the day. And I give the floor back to our host. Please, thank you so much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for our moderator, Mr. Andi Febrisianto, PhD. Ladies and gentlemen, we have now come to the end of our event today. Thank you for Mrs. Puji Lestari, PhD, Dr. Mulyana Hadi Pernata, Mr. Andi Febrisianto, PhD, and all participants. See you soon at the next sharing session. Thank you very much. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Terima kasih Bapak Ibu. Terima kasih.
Kabarnya gimana Bu Puji?
Baik Pak.
Selamat nih, Bu Puji.
Segera join nanti Insya Allah.
Ya.
Tinggal
kita rindu sama Bu Puji nih.
Pak Joni. Itu Pak Mulyana udah di sini. Pak Setiaji. Udah cepet. Tunggu aja Insya Allah sih. Udah sekedar proses.
Ingin dengar ceritanya Bu Puji.
Bu Puji.
Ini meeting lagi jam 11. Ini udah telat saya. Jam 11 sudah meeting lagi.
Ya.
Ini
Pamit ya, Prof.